Well, thank you very much to the organizers for inviting me today. You're going to know more about me than you ever would need to by the time this is done, because I'm going to spend some time going through my career. In preparing for this talk, I decided to Google Stanford. And it's a pretty good story, right? This guy, Stanford, was one of these entrepreneurs, a robber baron, built trains, grocery stores, and took his wealth, and one of the things he decided to do with it was build a university, which he modeled pretty much after Harvard with a focus on entrepreneurs. And as far as I could tell, this university spits out entrepreneurs like crazy. I think even Ron Davis's lab probably spits them out like pumpkin seeds in Half Moon Bay. It's just one company after another. And it's good at breeding these entrepreneurs. I'm going to tell you right now, I am not one of them. I've never started a company myself. I'm the guy the entrepreneur hires to actually make their dream work. So what that in the end means is I make other people rich. So I really am a lot like you in this room. I got an undergraduate degree at Penn State in microbiology, and then I went to Cornell and got my PhD in microbiology. I minored in biochemistry, genetics, and development. And then my wife had this fantastic job. I always wanted to go into industry. I never wanted to go into academia. Didn't think a postdoc was that necessary for industry at that time. While academia was getting to be now you had to do 2 postdocs, and now it feels like you have to do 3 postdocs. You basically spend half your life doing postdocs. So one of the tips I would start off with right now is I knew I wanted to go into industry, and back then it was not the thing to do. My committee did not like it. You should concentrate on being, the world's expert on, Bacillus subtilis gene regulation. Get an academic. They bred other academics, and it was discouraged. I was taking computer programming classes, things that were not part of my major. I just didn't need it. But in fact, in industry, you want to avoid specializing. You want to be an expert at everything, at least in the kind of industry that I wanted to be. You need to have a big toolkit for solving problems. And so I actually did my postdoc in something completely different. I went in and did a postdoc in mechanistic enzymology. Unfortunately, it was a lab, much like you, where they're concentrating on mechanistic enzymology. I am not doing synthetic chemistry and stuff like that. I cloned, sequenced, and overexpressed a bunch of genes. I knew how to do that really well. It guaranteed me a publication. And meanwhile, I brought in my toolkit, exactly what the heck what I bring to bear. For instance, a molecular biologist doesn't think about taking thin layer chromatography and analyzing their oligos that way for purities or nucleotides or stuff like that. But in fact, I do it all the time because it's just the easiest way to do it. But molecular biologists just have a completely different toolkit. And so one piece of advice is broaden your toolkit as much as you can. So my first job, the way I got my first job was I started applying. My first job I got from an advertisement in Science in the summer that I saw for Amersham, which is part of GE now. Amersham was a pretty big company. It was about 4,000 employees at that time. It was the first company that Maggie Thatcher privatized. They were originally like the radioactive guys for Europe. They did a lot of tritium, and they radio-labeled nucleotides, but they put the little radioactive dials on planes. I don't want to digress too much into that, but Amersham was a good company. It was a big company. I applied in the summer. I did not start my job until January 5th. It took them probably 4 months from when I sent in my application to even contact me. I interviewed around Thanksgiving and didn't start for over a month. So it takes a long time. I was hired. Let me see. I'm afraid that I got the list of questions yesterday. He sent it to me earlier, but I didn't read it until yesterday. So I changed my slides, so I'm going to screw myself up. Yeah, so set personal goals. I actually had a very few goals. I really recommend you do this. These were my goals at the time. As I said, I never wanted to go into academia. I wanted to make a product. I thought if I could make a product that made $1 million a year, I would sort of justify my existence. I know that tenure doesn't exist in industry, but if you make $1 million a year, you sort of have made like what you're going to make over your entire life for somebody. And I personally wanted to make $100,000 by the time I was 40. I'll reveal my salary information as we go along the way, because when I sat in your seat, that was the question I asked. How much do you make? And then I had kind of a higher sort of thing. How does life work? And I still kind of keep that. I like working in the biological tools industry. I never really like the data that people produce, but I love the technology that goes into producing it. Okay, so one of the things you got to start off when you work in the company is understand what the difference between where you are, academia, and industry. Industry is a very different environment. There's no tenure and stuff like that. The sword of Damocles hangs over your head all the time. Even though I've made, millions of dollars last year, if I don't make millions of dollars next year, I will be cut. And so you should be aware of that. So to put yourself in the seat of one of these entrepreneurs. When they hire you, they're not interested in bettering your life and stuff like that. They just want you to come to work and work, and be productive about stuff. They don't want your HR problems. Honestly, they just are trying to hire somebody who gets something done. They want you to create value. They're looking for compliance. This is an HR word that, I'm going to talk fast because I got a lot of slides. This is an HR word. I don't really like the word very much, but they use it all the time. And it basically just means like be professional, work together, get along. Don't put the company at risk. So by that I mean, don't sexually harass people, don't divulge company secrets, don't get arrested, don't violate other people's intellectual property and get the company sued out of existence. Basically just get along and get your work done. Now, if you're lucky, you work at a company that has some kind of higher missions, and I got to say, life is one of that. In fact, this one down here is actually one of our mission statements, understand how life works, which was one of my mission statements, so it kind of attracted me to life. But they do try to be a good member of society. We, have, one of these we produce no waste at the site, basically, and one of our missions is to improve and extend life. So it's nice to work for a company that has a higher mission, but most startups are really just trying to bring something new to market. My first 5 years at Amersham, so I was hired to develop DNA sequencing kits at their Chicago facility. I was hired by Rich Eichholz. As I said, I answered an advertisement in Science, and I was a stack of over 300 resumes. I was not the first choice. He interviewed somebody from Harvard who turned down the job, and I was his second choice. I fit a profile. He had written down, these are the characteristics. I want an Ivy League guy who has background in sequencing, blah, blah, blah. He had a set priority list, but I got to say, I think that because he came from Cornell, that helps. So I love this tradition you have. I noticed that it really is, it started a lot of people talking to each other and stuff like that. Networking is really key. The fact that you come from Stanford, I think, gives you a slight edge, right? Because there are lots of Stanford people in positions such as mine, and that little connection is going to help you. It's a little harder if you came from, Santa Clara or something like that. And I got to tell you, the culture shock of going into industry really nearly made me an alcoholic. My wife and I were probably closest to divorcing, and we've been married for 27 years. But that time was so bad. And one of the reasons is industry is very different socially from where you are, and I'm kind of I'm like those guys on Big Bang Theory. I'm not really good socially, and the thing is you stand up here, like all you guys stand up and you present your research, right? And you have been taught your entire time to try to prove your hypothesis wrong. That is part of your scientific training. And people stand up there, you've been working on this for years, and people shoot questions at you. Why did you do the experiment this way? Did you do the proper controls? Did you think about this other hypothesis? And you have to try to prove yourself wrong, and they attack you. And even though this is your life work, it's not personal. And then next week, you're going to sit down and you're going to sit in the audience, and you're going to do the same thing to somebody else. Well, I can guarantee you that most of the world considers that antisocial behavior. If a marketing person comes up to me and they hand me a document, they don't want me to question why it was structured this way. You know, they want me to they want me to check it for scientific accuracy. That's what I've learned. They really don't want me to do their job. And this grilling that I do to everybody around me generally is not really conducive to being well-liked and advancing within industry. So you have to control it and use it appropriately. And there were a number of social problems I had just adjusting to industry that way. And thank goodness for my boss. He really helped me through there. And then 10 months into my job, they closed down my site. Like they had me building sequencing kits, and then they weren't going to win. So they bought a company that had the best sequencing kit on the market, shut down my lab, and moved me to Shaker Heights outside Cleveland. And that was a great move, because that's where I really learned, I can't do that from here. That's where I really learned to develop products. So, these are two products here that were, probably got me off the ground. I worked on a lot of things, but one on here is thermosequinase. It was developed at Harvard University. The other one here, energy transfer primers and energy transfer technologies. These were developed at Berkeley. So again, I'm not the entrepreneur, I'm not the inventor, but I bring them to market. And the exercise of doing this, probably the 8 or 12 times I did it while I was at this company, taught me all of the development processes, from bringing a raw technology step by step through what they call a product development process to actually bringing something to market in a box, QC'd, it's going to work on your desk. There's a field called kitology. I haven't heard that word outside Amersham, but it's the science around building kits. And that is basically what I learned here. Okay, so here's a bunch of other. I'm going to skip through the science because I found from your questions that you mostly are interested in career guidance. But this guy was the first time I sort of worked. So I was mostly a reagent guy, enzymologist, molecular biologist. Then I started having to become an engineer because we basically bought 51% of a company called Molecular Dynamics down here in Sunnyvale. They packed my house and my wife and my family. I had a baby. There were 5 of us, moved, and they put us in a corporate apartment for 7 months in Sunnyvale. And I worked with my Molecular Dynamics, learning how to develop this instrument here. It was the first capillary electrophoresis machine on the market. And I gave several talks on that. And the nice thing here is now I become a systems guy. I'm not just, molecular biologist, enzymologist. I'm a guy who knows fluorescence, optics, all the various chemistries and engineering that kind of went into these things. And I saw all the mistakes we made along the way. And it was while I was managing reagent development that I got recruited by Mike and John Finney. So how did this happen? The energy transfer primers, I gave a talk at a conference, and at that conference, people see you. And one of the people who saw me was an entrepreneur. His name was Mike Finney. Thought I gave a good talk, thought I did good work, calls me up. I happened to be in Sunnyvale. He lived in the city, and I interviewed there. It was probably 5 months or 6 months after I gave a talk. And I wouldn't have answered his request, but I really kind of got stiffed from Amersham. So I started at Amersham making $45,000 a year. I probably was losing, because I had a family of 5, and I was the sole breadwinner in Chicago, I was probably losing about $1,000 a year, a month. I was in debt, probably $60,000, including credit cards tapped out. When I got my PhD, I had a child, and I was making $11,000 a year, and my daycare expenses were $7,000. So I was really pretty badly in the hole by the time I got mine, and I probably finished paying it off maybe 10 years ago. So when I left Amersham, I had launched all of these products. For the energy transfer work, I got a $2,000 bonus. I was not happy, so I was receptive when somebody came to me, and that was a wonderful move. So I spent 8 years with the MJ companies. I was hired to start a startup. I had a warehouse in Oyster Point in South San Francisco and a stack of about 300 resumes and nothing else. And so you start by building a lab. Fortunately, at Cornell, I'd already kind of done that. And hiring a staff. I hired 14 scientists, and we just did some amazing work at Bioworks. Probably the most famous work is fusion. The DNA polymerase, it is the best still, I don't know, it's been 15 years or so. It's still the best long PCR enzyme on the market by far. They hate me at Life Tech for inventing this guy because we don't have anything that can compare to it. And then it was, the technology behind that was a cover article in NAR. There's 2 technologies behind it. There's a long list of accomplishments here. I don't know whether I kind of go through it, so I'll go through it really quickly here. So we launched a couple of, Bioworks, this startup, was a research for hire company. We didn't have a license to PCR, but we developed the best PCR enzyme on the planet. So we licensed it to a company that did have a PCR. They basically grow up and isolate the protein, ship it to NEB, who actually sells it. So nobody's ever heard of MJ Bioworks. But in fact, it did a lot of really good work. And then I actually think there is a slide on this next one. I'm really distracted because I changed my slide so much. Oh, this is the technology behind fusion. Fusion is actually 2 technologies. There's this SSO7D is a protein that lives in sulfolobus, an organism that lives above the melting temperature of DNA. SSO7D's job is to keep DNA double-stranded above its melting temperature. It's a very abundant protein. It's a double-stranded binding protein, non-cooperative. It works as a monomer. And we found that, actually Yan Wang, who still works at Biorad, found that when you tether this to polymerases, you take a tack with a deletion of its exonuclease domain, and you basically over double its read length. This is a processivity assay. Only one binding event. How many nucleotides do you incorporate? Just a few here. But if you add this protein to it, you incorporate a whole bunch of nucleotides per binding event. So this is a process called processivity, and it did it with TAC, these are family A polymerases, and then it did it with PFU, which is a family B polymerase. And this makes sequencing much, much more efficient. Like I said, I'll zip through this. So the other invention is actually a polymerase evolution thing. So this is one of the things I do a lot. We didn't have a polymerase. I basically invented a way. We took one proprietary polymerase, another proprietary polymerase, and this is a way where we blended the 2 sequences to create a library with a lot of diversity. And then we screened from that library the enzymes that had the characteristics we wanted, and we combined them with SSO7D. And so here is one of these enzyme blends. This is a polymerase to exonuclease activity ratio. When you just take PFU and you just dump it into a PCR reaction, it chews up all your oligos because it's got all this proofreading activity. It's actually not that good a polymerase. It has some other characteristics that I do like, but this exonuclease activity is out of hand. So what people do is they just spike in a little bit into TAC or some other family A polymerase, a workhorse, and they get a blend. And so its polymerase to exonuclease activity is up here. PFU's down here, deep vent. These are the 2 enzymes I blended to make fusion. And then here's a bunch of the isolates from that library I was talked about. This is fusion right here. So basically, in this case, we're matching the polymerase to exonuclease activity, but it's first order instead of second order kinetics, which means the concentrations of the 2 proteins aren't important anymore. Right now, what you rely on is, in a blend, you rely on the editing polymerase to fix a mistake, and then TAC to come in and do the workhorse again. That's second order kinetics. It's actually third because of the DNA, but regardless, it's dependent upon the concentration of those things. Having it all in one molecule just makes it a heck of a lot better. And that's just, I've already bragged enough. So what MJ did next was they wanted to get into sequencing, and so they bought this company, a startup in Wisconsin called MJ Genesis. It was Genesis at the time, and they named it MJ Genesis for Mike and John. And we developed a DNA sequencer. It was a slag gel electrophoresis sequencer. It did 96 samples at a time. It was really great. I love this machine. We sold about 50. And the reason why we only sold 50 is because capillaries came out at the same time. The guys that I was working on before brought the instrument to market, and they kicked our ass. So what I did, they made me president and CEO of this company. So I was still running Bioworks, and now I was president of a company. I had about 45 employees at the time. And we changed. When the sequencer didn't do too well, I turned around and asked that to become the site of real-time PCR, quantitative PCR. And this is one of the instruments. This has everybody know what an MJ thermal cycler is? The little black boxes with blue handles, they're still around all over the place. I've got a couple in my lab. So there's a cycler inside this, and above it is a PMT, a photomultiplier tube to do detection, and then an array of 96 LEDs that light up each well in a 96 well plate individually at a time to do detection. It's a really nice instrument. And so this site became a manufacturing site. So suddenly I was manufacturing about $20 million worth of these machines a year. And that was going so successfully, MJ asked me to actually become head of engineering for the entire company. That happened because they launched a machine called the Dyad. They shipped 115 of these instruments, and 100 of them came back. So it launched late and broken. So they fired that engineer, and they made me head of it. And so this stuff is pre-me, and this stuff is after me. And you know, robotic lid was the first thing I kind of came out with, and probably, I don't know, probably 7 or 8 thermal cyclers while I was there. And then this box here is one of the ones I'm most proud of. This is actually a 4-color real-time PCR machine. There's an XY stage inside here. This is a regular thermal cycler. These are these alphas that are exchangeable on MJ machines. And this tiny little die about that big that you can switch which 4 colors you want. And it moves around on an XYZ stage and XY stage, no Z. And it does 4-color real-time PCR. Great instrument. I have one in my office. I'm so proud of it. We also developed these 2 instruments before Biorad bought MJ, but they launched only under the Biorad label. I will say that I was the first employee that Biorad fired after the merger. Probably the reason for this is I loved MJ. I liked Mike and John Finney a lot. I felt very indebted to them, and I was militantly in their camp during negotiations. So I was unemployed. It probably took me, I got, so when I left MJ, I think I was making about just over $200,000 a year. And it took me maybe 4 months, almost 5 months to find another job. I did it mostly by networking with people that I had known before. Of course, did all of the ads, but mostly it turned out to be networking with people I knew before. The guy who was the chief science officer at Molecular Dynamics, who I had collaborated with at Amersham, I had kept in contact with him over the years. And that company, Molecular Dynamics, was rolled up into GE. And that group of people, the CEO and all of the intelligence behind that company, moved to San Diego and started a company called Illumina, which some of you may have heard of. So I was still in contact with all my old friends. And the chief science officer basically got me an interview at Illumina and an interview at this startup, which at the time was called Cell Biosciences. So there's a list of accomplishments up there. Basically, I led the team from feasibility. It's a long story. It's a wonderful story. I basically started a collaboration with a guy at Berkeley to look at this technology, and they turned around and bought the machine for $400,000. We then turned around and used that in a round of financing. We had a nice up round. The company was formed on $14 million. We had an up round of $28 million. And like I said, I was made chief operating officer of that company. We'll go through the tech. So the black box here, the one that looks like an MJ machine, is the one that I sold. And then the company, when I left, changed its name to ProteinSimple, and these are the boxes that they sell now. Quick explanation of the technology. What they do is they've automated the Western blot. Everybody knows what a Western blot is? They automated it. They take a 5-centimeter capillary. We coated it with some cool chemistry on the inside wall, figured out how to manufacture it in 50-yard rolls of capillaries. What you do is you take this capillary, and in that machine, it's all automated. You put in a 96-well plate, you push a button, you walk away. It will fill these 96 capillaries with the contents of cells. You know, and it was pretty sensitive technology. We got down to the PNS paper was 25 cells worth of cellular content. But when we just measure ERK, we can detect ERK down to 1/3 of a cell. So it's very sensitive. You fill the capillary with the cell. You do a focusing reaction. Oh, it's going to go through the whole thing, huh? You focus your proteins of interest. And in this case, I'm doing isoelectric focusing. So it's separating by its isoelectric focusing point. When you add a phosphate, you change where that point is so you can separate phosphoisoforms of the proteins. You then shine this light on it at the right wavelength, and and the chemistry on the walls of the capillary gets activated. It captures all the proteins at their position. You then wash out the tube, and the proteins are stuck in place. So then you can come in with primary, secondary antibody, just like normal, and you can visualize it with HRP. You take a picture of it, and you get these bands. And what can you do with this? Well, you can use different antibodies to figure out which band is which is a single phosphoform, which is not, and you can start assigning the phosphoisoforms of all the proteins. And ERK is a key cell signaling protein. So people are interested in taking these and doing all sorts of experiments looking at cell signaling regulation. So it's kind of cool. Couldn't do anything. It's my little foray into cell biology. And then of course, the system works in size separation as well. So automated Westerns, which is pretty cool. Okay, so there comes a time in startups where the entrepreneurial CEO is not always the best person to run the company. This is not actually an uncommon thing. Even Steve Jobs was fired. These are very often flamboyant personalities. And very often they're a little eccentric. And that's what gives them the courage and the imagination, I suppose, to actually decide to take on one of these sorts of things. Well, our CEO was one of those people. And eventually the board of directors let her go. And they hired a new CEO who brought in his own team. And so I was let go. I was making, I think, about $230,000 when I was let go from that company. Can't tell you any more about my salary because I'm still working at Life. And I don't want to divulge any confidential information. So my, I was looking for, I didn't quite expect to get fired, but I now began to search for another job. And I made my job of getting a job very serious this time, much more seriously than I had in the past. It's hard to replace a $200,000 income. And so I applied a lot of places, but I went to war with Life Technologies. I decided that was the company. It was a tool space that I was very comfortable with. I think I could deliver a lot of value for them, so I targeted them. I was not in a happy place after I left Cell Bio. I was kind of depressed. I was willing to take a job as a mailman. I was not going to be all ambitious anymore. And I actually applied for a junior enzymology, just wanted to go back to the bench, not have to deal with, I ran 2 HR departments, and the worst job of being a boss is really managing people's HR issues. And with a crazy CEO, that was 80% of my time. I just wanted to bring products to market. That's my brand. I bring products to market. And I didn't want to worry about manufacturing and crap like that anymore. So I applied for a junior position, and I also applied at Illumina, and Life didn't listen to me, and they started competing, and I ended up getting a pretty good package, but it wasn't going to be as a junior benchtop person. They wanted me to be the architect of a single molecule sequencing platform. And so nobody does single molecule sequencing without showing a movie. And this is the quickest way to actually explain this technology. It starts with a microscope. You might want to turn off the lights because it gets really dark because we turn off the lights. Let me pause. Okay, so we haven't turned on the laser yet. Oh, you can't see anything. I see what I did. Sorry. Okay, you can probably just make out here. What we're looking at is a slide down here. It has an oligonucleotide attached to it. And then annealed to that an oligonucleotide is this long piece of DNA. I did it again. This is what happens when you don't practice your talks. Okay, so in flies this guy right here. So what are we looking at here? This is a polymerase molecule, and attached to it is a quantum dot. So a quantum dot is basically a piece of semiconductor. It's a semiconductor crystal that fluoresces. It fluoresces the wavelength that you can select it. It's based upon the size of the crystal itself. I won't go into that chemistry, but it's pretty cool. So it's a very, very strong, very high quantum yield, very, very strong, basically dye. It's stronger than most dyes you could possibly think of. Okay, so the polymerase binds at the 3' end right here, and then we turn on the laser light. And of course, the quantum dot glows, and so you see these arrays now of DNAs. And then what we do is we add some nucleotide, but the nucleotide on the phosphate end, on the terminal phosphate, has a dye attached to it. So what happens is the polymerase starts sampling for the correct base, and when it finds it, it binds. The quantum dot excites it, and it glows. So here it is coming in. It's finding this is the correct one. It's going to dwell there for a little while. The quantum dot, the fluorescence from the quantum dot, is exciting the dye on the end of the phosphate. Here's the phosphate chain. This is where you're going to get the phosphate transferring. Here's the base paired up, and the break is going to happen right here. And then the rest of the phosphates, because you're only putting one into the chain, the rest of the phosphates with the dye attached are going to diffuse away. Okay, so you're just going to see, watch right here, you're going to see the little chemistry happen right here, and then the dye is going to just zip away. There it goes. Now the correct base takes a long time, and the incorrect base is on a much faster timeline. So you can see that's the correct one, you get a glow, it moves to the next position, it's going to sample. Nope, that's not right. And then this is all happening on the slide individually. So you're looking at each individual molecule blinking at you. And then these dyes are completely invisible until they bind to the close proximity to this dot. And so you can start doing 4-color sequencing. Actually, you can do 5 because the quantum dot has a signal that drops when you do energy transfer. So you saw this blue dye go in, this T, and now we're going to have another base come up. It's an A. You see the A signal go up, and the quantum dot signal goes down. So that's confirmation of a real event. And signal to noise is the name of the game in single molecule. And so in this way, you can do 4-color sequencing. I got to tell you, this was the coolest project to work on. I mean, everything comes together. Optics, fluorescence chemistry, enzymology, it's material science in the quantum dot, it's the software. I mean, this is where I live. I love where these complicated things involving a lot of different technologies and expertises all come together. And if you're a molecular biologist, you kind of represent the end user. You're actually one of the best type of people to be involved in a program like this, because the engineer doesn't know what good data looks like. They know a spec. But you as a molecular biologist, you're kind of the customer. And so you're actually in a good position if you're a molecular biologist or have that kind of background to be able to help out with something like this. So I just love this project. The unfortunate thing about this project is the machine that does it costs about $600,000. And the accuracy that we achieved was just south of 90% accuracy. And if you're doing sequencing, accuracy is the name of the game. And there is a market for 10 KB reads of 90% accurate, but it doesn't necessarily justify a $600,000 instrument. So Life Technologies came to the conclusion, and we had a competitor, PacBio, which is in Menlo Park, was our competitor in the same space. We were playing catch up with them. We thought we could win, though they were spending a lot more money than us. It's just that we were all fighting over what would actually did turn out to be a very small market. It wasn't worth the investment. So they turned around and bought a company called Ion Torrent that they did think was an investment. And they quickly asked me to be head of sequencing biochemistry for that project. This is a launch product now. They had just launched it when we bought them. And so this is sequencing on a semiconductor. Here's the two chips. These are marketing slides, right? PGM has throughput that's suitable for looking at individual genes, and the Proton has throughput for looking at individual genomes. We'll talk about the technology real quick. It's basically a pH meter. It's not that fancy, right? You take a bead coated with the DNA you're interested in, and you put it inside a pH meter, and then you flow nucleotides across it one at a time. And if it's the correct base, you're going to produce a little acid, and that circuit inside the computer chip actually detects that acid. And so if you flow an A across and you get a signal, it's an A. If you flow a T across and you don't get a signal, it's not a T. Now, the key is that, a chip, can pass these around. This is what the chips look like. And so the smallest chip has 1.1 million pH sensors on it. That's the smallest chip. That's the one they launched when I joined, and our read length at that time was about 50 bases, which 50 bases, 1,050 bases is actually pretty useful if they're accurate. What my team's job was to get longer and better. So we did an enzyme evolution project. We invented new chemistries. My team, I run sequencing biochemistry. We basically were very quickly sequencing 600 bases long. It's not unusual for us to sequence 600 bases long. Right now, commercially, I think our kit is around 400. So what we do in our lab and what we do commercially are a little different still, but that's where we're heading. And so here's the size of a well and the signal you get from one of these chips that have a million sensors. The next chip came out, had 6 million sensors. And then the next chip came out, had 12 million sensors. Okay, so not only are we getting longer, we're getting way more dense data. So now every single run we look at 1.5 gigs from our 318 chip, the smaller chip there, 1.5 gigs of data, and we go, what a crappy run. Our records are over 3 gigs now on that chip. Well, the other chip that I've handed out there is this Proton 1 chip. That has, so we're at 12 million. That chip has 160 million pH sensors. It's amazing. And really the signal does not look that different. Everything's scaled down. We shrunk down everything, shrunk down beautiful. And right now in development, we have a chip that has 660 million sensors. So when that guy's doing 100 bases long and it's 660 million sensors, we're basically going to sequence your entire genome in just about 2 hours, at least to do the run. It'll probably take all night to analyze the data, for about $1,000. And when you get down to the $1,000 genome, now you're in a different land. Now babies are born, their heels are pricked, you sequence their DNA, you hand mom and dad a little thumb drive with their kids' sequence, and it is used to inform and extend their life for their entire life. They get high blood pressure, you choose which, by then we'll be able to choose which drugs are appropriate to give that child. So this smaller machine is still useful for individual genes. You have breast cancer, just test the breast cancer genes. We have kits that basically enable that. If you don't know what's going on, we can sequence the entire exome on this P1 chip, and when the P2 chip is commercially launched, we'll be sequencing entire genomes. Pretty cool. But cool technologies don't sell products. You need the application. And for P1, it's just this, we basically do 2 exomes on this, on a P1 chip. That is, 95% of all the interesting genes currently that people have identified are in exomes. So it's that and the kits that making the library that are going to sell the instrument. The technology is cool. Nobody buys technologies. They buy applications. Okay, So let's go, from here on in, I'm just going to do career development stuff. So like I said before, let's make sure you know what your goals are, and then develop a strategy to sort of achieve them. And like I said, I kind of went to war. So you have to make some decisions, okay? Are you willing to move? This is my path. That's a lot of moving for a family of 5. Most of them were company moves, which are pretty good. I mean, the packages for life was a very generous one for an executive. Most of them, like a postdoc, we probably hand out. It depends on the company and stuff, but it's not unusual for us to say, here's $5,000. We'll see you in San Diego. In some cases, in higher levels, we move people all the time. Are you willing to travel? My job at MJ Research, well, here's where I was living. MJ Genesis was here, where I was developing the sequencer and the real-time instruments. And MJ Research, all the engineers developing the thermocyclers, were over here in Boston. So 2 weeks in San Francisco, Sunday night, put my kid to bed, red-eye to Boston, at work Monday morning, Wednesday night, fly to Chicago, Madison, drive, late at night I arrived there. Thursday morning I'm at work, my office in Madison, Wisconsin. And then Friday night I'm hopping a plane to get back to my family. 2 weeks home, 1 week on the road for about 4 or 5 years. That's a lot of traveling. Now I'll just say that moving and my willingness to travel actually helped me move up a lot faster. Because if you're willing to make sacrifices for your company, that generally enables you to do more things for the company. You kind of want to think about the small, medium, large thing about companies. They're really, really different, okay? So a small company like Bioworks was, or GeneWorks was, or Cell Biosciences was, resources are very limited. You don't have a lot of money. You know, it's like being in a tenured professor's lab. You're building stuff yourself. You're not quite blowing your own glass, but you're not ordering, you know, gene synthesis from scratch. You usually only have one program because that's all you have resources for. 40 people focused on one thing. Everyone does everything. The CEO takes out the trash if that's what's needed, right? The role models and experience are generally lacking. My CEO was a little kooky. She was not a good role model for the people there who wanted to be good managers. She was not the person to look for and study. And like I said, the cultures can be extreme. So a lot is driven by what the leadership looks like. If the leadership is a crazy person, then you're dealing with crazy. If the leadership is you know, you're going to work 70 hours a week, you know, that's what that's going to be. If it's another one might be all laid back and, you know, we're going to play ping pong at lunch. But there's lots of promotion, right? Because life is tenuous. They got to keep people there and working on this. A lot of times these companies go through highs and lows and you feel insecure, like maybe the company won't make it. In fact, 9 out of 10 of these companies do not make it. So you almost go into it thinking like, okay, yeah, I want a lot of stock, but chances are that stock isn't going to be worth. I still have, I don't know, a couple 100,000 shares in ProteinSimple. If they ever go public or sell, I'll get some money. But who knows? It might not ever go to anything. And since they don't have a lot of money, they promote fast. It's not hard to become a VP relatively quickly at a small startup because if they like you and they really think you're good, they might not have money, but they have titles, and titles are cheap. However, a VP at Cell Biosciences is not a VP at Life Technologies. So your title may not be transferable, but you shouldn't be thinking about that anyway, and we'll talk about that later. And like I said, this is the brass ring. I was young enough that I thought I can take one more stab at the stressful existence it takes to try to get rich. And it didn't work. All right, so medium, MJ Research. This was my sweet spot. Certainly I would describe MJ Research as the place where I moved up the most. I was given a lot of opportunities by those guys. But it really depends on the ownership. They were a very benevolent dictatorship. They were very benevolent to me. But I'll tell you, we fired 5% of our staff every year. That's just a rank and yank system. If you were good, you stayed. If you weren't good, we got rid of you. So if you're good, you had a good job, and they partied and had a good time. It was a very relaxed environment. There was a keg in the beer fridge. It was fun. I really liked working there. If you're lucky, those companies are growing. When I joined MJ Research, they were about $25 million a year. When I left, when the company was sold, it was just a hair under $100 million. Growing pains mean by the time we moved into a facility, we had outgrown it, and we're looking for another facility. PhDs that we hired as sales reps became order takers because the instruments were so popular. They just didn't have to sell. And we became an R&D driven company, which is not a good thing. You should be a market driven company, which means marketing should be leading more. But the technology was so good and the instruments were so good, it was just fun. Of course, when MJ actually lost a lawsuit on PCR, and they no longer could sell their instruments. We were in bankruptcy. We declared bankruptcy so that they couldn't actually force this on us. So we had this successful launch of the chromophore I showed before, most successful launch in the company's history while we were bankrupt. I quite literally, part of your job as boss is to smile and like, we're gonna make it. But I would walk into the bathroom, splash water in my face, look up in the mirror and say, okay, I can do this. It was very much trying to psych myself up so that I can go out and be, this cheerleader role, like we're going to do this. And we did this very successful launch. It enabled MJ to get a better offering for the company. So it was good, but shrinking or that kind of stress, if the company is going on the downside, is not fun at any company. And then there's the large companies. My two experiences are Amersham and Life Tech. I really like both these companies. I got formal training at Amersham that really helped me get going. I had mentors there that were excellent examples. Not necessarily my bosses in particular, but they recognized that I worked hard. A couple of them took me under their wing. I was kind of fast-tracked. It really at a minimum, I got to work on the most interesting projects. But it gave me formal training. They, taught me what exactly sexual harassment is. These are good things. How to actually develop a product. They had a formal product development process that was written down, step by step, how to do it. And in large companies, they specialize. It's the opposite of the CEO taking the garbage out. You do your job, and you hand things to other people. It's specialization is how most of America has advanced. Is because everybody does very narrow things. You don't make your own clothes. 100 years ago, you had to make your own clothes. So specialization. Everybody is just more sane. I mean, they're just not crazy people the way you can at other companies. You can have these flamboyant personalities. Those people don't last very long. Life Technologies, actually both of these companies, get ready for a drug test. Get ready for a background check. I mean, they went all the way back to New Jersey when I was a minor. They will check you, and if you're in any kind of a sensitive position and you have a criminal background, not in all cases, but in some cases, it'll prevent you from getting a job. All right, and very abundant resources. If you're working on a high priority project, if you're not working on a high priority project, they're sucking resources into that high priority project. If you see a high priority project in a big company, try to get on it. That's where the action is. That's where the fun is. You want to be associated with these things. And it's fun, right? You have the resources to get stuff done, and you get stuff done. Okay, so finding a job. So as I've already said, finding a job at our level takes a long time. My first job, I said, I applied in the summer. By the fall, my wife was pregnant with our second child. I was churning out 10 CVs a week. And I'll talk about CVs later. Now, the higher up you are, the more time it takes. That is, it's harder to, not a lot of $200,000 a year salaries come up. Right? It's just not that common. So you're kind of waiting for one of these positions to open up before you fill it. So the higher you are, the more you that's why when VPs are asked to resign, they get a half a year severance. Because it's going to be a long time before that man or woman gets a job. By the way, typical severances are 1 to 2 weeks for every year you've worked for the company. Okay. So a startup, and I used to be great at this. When somebody walked in and they were good, I would stall them somewhere. I had a process. First person you interview is my lowest person in the lab. And you just kind of go up. And as you go up, if you reach me, you're golden. Because I don't want to waste time interviewing just everybody. I'm busy. So if you reach me and you're still in my office, congratulations, you're in pretty good shape. In a startup, if I really liked you, I would pull a couple of people. You'd be interviewing my receptionist for a little while, and I would be in the back room with 3 or 4 of my key team members, my decision makers, and we would try to put an offer together for you before you left the building. That's not the way it works at a big company. Big company, once you get, I don't know, where did you start here? You interviewed how long ago? 4 months ago. You still don't have a written offer, do you? Okay. So we still don't have a written offer. It really does take a long time for large companies to kind of get to, even with a verbal offer, we're basically talking about the background checks, the drug test, all the various things. You have an immigration issue. Hope you don't mind me pointing these things out. But these things take a lot longer in a big company. I need 5 signatures just to change my thing. If I got a scientist level 3 and you come in and you're a scientist level 2, lower, you think it would be a no-brainer. I still got to go back and get 5 signatures, even though I'm spending less money. And if I got to go higher, it's going to take me a week to get those 5 signatures. And, crazy. But that's what happens when you're in a big company. They've got lots of checks and balances. So it takes a long time from the interview before the company actually will probably hire you, a couple months. So the CV gets the interview. So you got anybody here actually getting advice on writing CVs? Anybody? Okay. So typically, I'm going to spend some time on CVs because I read a lot of CVs. And I've given this talk, talks like this at a number of places. And I cannot tell you the number of times a postdoc hands me a 5-page PhD, a 5-page CV. You know what I do with that? I throw it in the garbage. I don't have time to go through every single experiment that you've done. If you think every single thing on there is really so important that it's going to get you the job, you're dreaming, because nobody's going to read it. In fact, the first person who reads your CV at Life Tech is not a scientist. They're matching a spec to a document. And if they have to search through that document, they got 299 others. They're not going to do it. So let's talk about CV on the next one a little bit. So the hiring manager has a large stack of resumes. So they state, right? You want to develop a narrative that every aspect of it is key to getting, like when somebody looks at it, they kind of figure you out. And you get to choose what that narrative is. And to be honest, I have more than one, So I have an operations side and I have an R&D side. And I will tailor my narrative to the job in the company that I go to. Okay? So I customize every submission. That's why I only can do 10 a week. Because I'm really working hard on making sure that I hit on that. Okay? The CV gets you the interview gets you the job. Again, study, like this is the most important thing you've ever done. There's nothing worse than a postdoc coming in and he doesn't know what the heck's going on. We tell you the list of who you're going to interview with at Life. Google every one of those people. You should know what they've done. They might be working on the same project that you worked on last year. And in fact, that happened to me at Cell Biosciences. Googled the guy. Turns out my undergraduate work was very similar to his when he was a professor at Stanford. And I don't let him know I Googled him. I just, go through my history and I worked on attenuation, yeah. And, he's like, really? And he starts drilling me on attenuation. And I know it. Because I knew beforehand, I set it up, right? I knew that was going to happen. Google, if you know who's going to interview, Google them, know them, and figure out some way to make a connection with that person. Okay, so study for your job, and please practice interviewing. I've got about 20 questions that I ask. Most of them you can find in a book or online. Okay, I try to get people off. I make it a mission to get people off of their script. So I'll diverge from that. But you damn well better be able to answer, the top 100 most likely questions that you're going to be asked. You better be prepared. When I ask you, what do you want to do in 5 years? Have an answer. It may not even be the truth, but, have a vision of where you're going with yourself, right? Okay, so practice. Okay, and don't leave your postdoc until you have a job. Whenever you have a chance, it's much harder to find a job when you don't have a job. People are going to ask, why don't you have a job? And if you don't have a job for a year, they're going to start thinking there's a reason why you don't have a job a year. There's two possible reasons. One, you interview horribly, or two, you're too lazy to put out CVs or something. Or maybe you have a good narrative. I'm a mom. I took a break to have a child. I'm ready to get back into my field. It's a great narrative, right? But you darn well, if you're out of the workspace for a year, you better have a reason for it. By the way, don't jump jobs too often either. I start looking at people and wondering, what's going on there. You probably can't read all this, can you? Okay, so this is my CV. There's one other page that's just a list of references and patents and stuff like that. I don't generally include that one unless people ask. At your stage, it's probably important to be well published. At my stage, nobody cares how many publications I have. They care more about my patents. So right at the top is my contact information. And then I don't write an objective. The job I'm looking for is my objective. In this case, this is the one I used this is the resume that got me the job at Life Tech. So it's a 4-year-old resume or whatever. So my objective was I was a senior R&D executive. In other words, in other cases, I was a senior operations executive. It depends on where I was applying and what I wanted to do. Each company that read my resume saw a slightly different bend. Okay, so 16 years in the research, development, manufacturing, and launch of many multi-million-dollar a year commercial products. What did I say a company wants from you? A company wants to make money. That's why they exist. They want to increase value of the company, increase intellectual property, whatever. They want you to come invent or produce products for sale. Okay, so I have a record here of actually doing that. So that's what I'm going to put right at the top. That's what companies want to do. That's what's right at the top. So I have like these 6 bullet points that are like that. They're basically why you should hire me in 6 bullet points. An executive summary of my strongest characteristics. Okay? Most people are going to stop right there, right? The person who reads your resume first is some HR talent person who doesn't know good science from bad. But they can read this whole section just fine. They got a job for a senior R&D exec. There it is right there. It's not, I want to run my own lab in 5 years. I don't hire a person who wants to run their own lab in 5 years. I'm interested in a person who wants to bring products to market. Right? And that's right there at the top, your strong suits. I'm an expert at cell biology, maybe it's a particular pathway at your stage or stuff like that. You could be the world's expert in some pharmaceutical target or something like that. It's amazing the number of resumes I read where on halfway down page 2 is the skill that I'm interested in. Did they not read the job description? Did they not put that skill near the top so that even an HR talent person can see right from the beginning that this is what Peter Vander Horn's looking for in a person? Get it right out there. And then I follow up with employment. So here's 1 job, 2 jobs, 3 jobs. And then my education down here, numerous presentations, scientific conferences, a couple of boards of directors. Invented fusion jumps out because I'm very proud of that. People know what fusion is. Nobody will remember who Peter Vander Horn is, but a lot of people know what fusion is. So see how it just jumps out? So I spent a lot of time, probably 2 or 3 weeks working on this document to get it really right. 2 pages, not 5 pages. And I have a lot of accomplishments. If it was longer, it won't get read. Once it gets from that HR person to the hiring manager, that's a scientist, then they're going to read through this in detail. And that narrative is all going to be consistent. This is a product development guy. He knows how to do that. Okay. I told somebody, gave this talk at Scripps, and a person sends me her 5-page CV, and I told her, you cannot do this. But the career person said you had to put everything in there because there'll be computer searches for those words and stuff. It doesn't take 5 pages, please. If you can't, especially at your stage in the career, 2 pages is probably plenty. It is plenty. Any questions on CVs? Okay, salary. So I already gave you all the salary information I'm allowed. So negotiating salaries, this whole web page is on this. I never tell people what I want in a salary. I only tell them what my last job did. And you can do the same thing. Look, I'm a postdoc. I make $21,000 a year. That's what I made. And that's all I really need to know, right? Most people have a spec. They want to know what's the lowest amount they can pay you and get you to join. And you want them to give you the highest amount they can give you. So the first person who talks, at least the feeling is, and probably is true, the first person who talks loses. But there's nothing wrong with telling them, you know, I make $30,000 a year. I'm a postdoc. Okay, and your first job, like I said, I was losing money in my first job, but it was such a perfect job. Hey, come develop DNA sequencing kits for us. So I'll take anything. I just want to get in. And then once I get in, now I have the opportunity to prove what I am, what I can do, and that salary goes up pretty darn quick. Most companies are pretty good at rewarding people, and if they don't, then do what I did at Amersham and jump. Once you're in. So first day at work, I cannot tell you the number of people who walk in, and they're learning about the technology, and you know, they're really, really smart people, and they know how to fix it. They know how you shouldn't be doing it that way. And that's fine. I love the eagerness and everything, but before you start making, you know, suggestions upon, there's probably a reason why we're doing it that way. We're smart too. So I try to go as long as 6 months without trying to change the direction of the company. So just remember, you're smart people. You have plenty of time to demonstrate how smart you are. Just use those times, not your first week on the job. Big mistake I made at the company that I said my first 6 months was horrible was, you know, you guys are doing stuff wrong. Bad idea. Okay, so here's the keys to success. Intelligence, attitude. Now, everybody here, they have PhDs. That puts you in the top 1% of the entire world's population. And at Stanford, that puts you in the top 0.1% of the world's population. So let's stipulate that you guys are amazingly smart, okay? After that, it's attitude. You know, it's all how you're going to use it. What are your goals? Are you going to come in and work 9 to 5, or are you going to come in and do, you know, 9 to 5 plus extra? You know, are you going to do that little extra? Are you going to show me that you're excited about the job? You're going to, it's your attitude. You want to be successful. If you're just kind of like going through the motions, that's fine. There's plenty of PhDs that hide in big companies for a large part of their lives, collecting a salary, getting their 2 or 3% raise a year, and that's a perfectly fine existence. But if you're at Stanford, you're probably relatively ambitious. You may not be, you know, off to become the next I don't know, Steve Jobs or the Google guys, but you know, you're probably pretty ambitious. So think about your attitude. Get excited about your work and express. I mean, you can see I exude excitement. I mean, this is, if anything, I'm over the top. Okay, and then just do this little extra. You know, is there anything else I can do before I go home? You know, I learned that in the restaurant industry. You just go up. Is there anything else I can do before I go home? And they love you. Nobody ever asks that. Okay. Every presentation is an opportunity. Remember, I got my job at MJ because of a presentation I gave at a scientific conference. okay? But it's not just that. Even within Amersham, many times I was asked to give presentations to board members. You know, as a junior guy, giving a presentation to a board member. Those are really, really important presentations. You should become really good at PowerPoint and really good at standing up here like I am and making a connection to people. Don't just talk to one person. You see me, I look around the room, I present, right? You be good at that because it actually, I mean, it will move you up much faster if you can address it, especially executives, you know. There's a reason why the executive summary is a bunch of bullet points. How to get to what is important to the executives and deliver that message clearly and succinctly so that they remember it. It's really, really important. Know your audience, how to speak to them, do something extra. And then develop a brand. I kind of told you a little bit about my brand. That's something you have to kind of decide for yourself. What is the image that you want to project of yourself, you know? And everybody kind of has a brand. And as a manager, I kind of make it a mission to understand my employees' brands, you know, to understand, okay, what do you want to do 5 years from now? What's your life's goals, you know? And then the key as a manager is to find, okay, here's the company's goals. Here's that person's goals. I want to find the overlap, the commonality between the company's and the employees' goals, because that's where I get the highest productivity and the people really excited about doing their job every day, right? So develop a brand and let people know it. By the way, all this is easier for extroverts than introverts. Okay. Every day, I have 17 employees right now. I touch base with them every day, and most often I'll walk them, what are you working on? What are you working on today? Okay? And they know now, after about one day, that they should be able to tell me what they're working on and why it's important, just every single time. And it's not that hard, just be ready for it. You know, if you're mixing up some little reagent, you know, I'm testing the effect of magnesium on this polymerase. I'm optimizing it so that it can go into that kit next month. You know, just a little blurb like that. Always be ready with your executive pitch, okay? Avoid becoming a specialist. And I know in academia, there's a big difference between academia and I mean, you in academia want to be the world's expert on, you know, gene regulation in Bacillus subtilis, you know? That's what you are. But me, you know, I know material science, I know electronics, software, I'm a programmer, I'm a molecular biologist, I'm an enzymologist, right? It's great to have as broad a tool chest as possible. And if there's anything that separates a PhD from everybody else out there is that in 6 months, you can become anything you want to be. If I told you you need to learn electrical engineering and program this PIC chip in 9 months, and you didn't know what a PIC chip was, I bet you in 4 months you could do it, if not less, because PIC chips are actually pretty easy to program. But I'm just telling you, the thing about being a PhD is you are courageous in going into the unknown and learning something completely new and becoming an expert at it. That's what we've taught you to be, right? Or what the schools taught you to be anyway. And that's very different from, you know, people who are college grads where they learn textbook knowledge. They didn't actually have to do it themselves, and it's a long time before they have that confidence. So that's your secret power. And resist, you know, getting pigeonholed as the person who is, you know, great at one assay. You're great at one assay, but man, don't become the only person in the company who's great at purifying a particular protein. You want to be the person who purifies all proteins, and you know, can also do enzymatic kinetic assays on them or whatever. You know, you can do Michaelis-Menten curves or whatever the heck you need to do for it. And the great thing here is, like I said before, in industry, you have to solve problems to bring things to market. And the bigger your tool chest is, the more you'll have to bring to bear on problems, okay? If you know MATLAB, it's just amazingly nice to know MATLAB. Very few people who are molecular biologists know it, and it's just so useful, like over and over again I see in careers. You can learn MATLAB in 6 months, easy. Okay. When people come to me with problems, I need to know problems. Don't hide problems. Don't just come to me with problems. Come to me with problems. I've got like 10 minutes to deal with this, okay? Come to me with problems and a menu of solutions. I may pick another one, but I can't just deal with just people throwing problems at me. So give me a menu of solutions as well. I'll tell you one of the things it's good to do in a company is turn something around. So the energy transfer project, after I got a bit of a name for myself doing the thermal synchronase work, my boss was not somebody you would use as a mentor, kind of assigned me to this project called Energy Transfer Project. He told me at the start, this product is only good with TAC. It's only really needed for TAC. Thermal synchronase doesn't need it, okay? I was the only person assigned to it. Turned it around. The stuff I made between Thanksgiving and Christmas sold in my beta test for $4 million. I mean, you just completely wrong. Turn something around. The engineers with the failed Dyad launch compared to the launch of the chromophore. Turn something around. Don't be afraid when somebody hands you a dead-end project, especially since you can go home, dream about it for a couple of days, you may be able to figure out a way to make that into a winner. And when you do that, that gives you a reputation. Okay, avoid commiseration clubs, especially when things are going down, and things do go down in companies for long periods of time. You know, people get together and they start talking, right? These are very destructive. If you think about a team, you know, if you pop fly out to the pitcher or whatever with the bases loaded, the last thing you want when you go back to the dugout, the last thing that's helpful is a bunch of people whispering in the corner about, you know, what a crappy job you did, right? You're working on a team, break up these clubs, don't partake in them. Yes, we know things aren't going well. We have to acknowledge them, of course, but people sitting around pointing fingers at managers and stuff like that, it's just really, really destructive. So, and you get associated as one of those kinds of people, it's just not good. It's not going to help you in your career. Okay. Try to do your boss's job. So this is a funny one, right? Because I heard this from Amersham the first time. I didn't hear it again until I was at Life. Jonathan Rothberg says this all the time. And people laugh because it's such a weird thing to say. Try to do your boss's job. I didn't say try to take your boss's job. Try to do your boss's job. I'm working hard. You know, if you can make my life a little easier, taking some responsibility away from me, I can now work on taking my boss's responsibilities, right? I become more valuable to my boss the more of his responsibilities I take, and he is now free to work on his boss's responsibilities. So, you know, get your job done efficiently. If you can get it all done in 8 hours, more power to you. If you feel like you want to be one of those people, and you want to work 10 hours or 12 hours, then, you know, walk into your boss. Is there anything I can help you with? As he or she gets confidence in your ability to do, they'll get more and more responsibility. And guess what? When he gets a promotion, guess who's in line? Okay, always try to work on something important. I think that's pretty obvious. You know, in life, there's hundreds of projects, and we all know who the winners and losers are. And, you know, there's a couple of people on my team now, they were very, very effective at getting moved on to my project. Because, and their project is shrinking. So move around. It's good in companies to move around within projects. Move laterally. Don't expect a promotion or anything. Excuse me, because it is just a heck of a lot more fun to work on you know, the stuff that's important to the company than it is stuff that isn't. Okay. And then be careful what you ask for. So really, like I said at Cell Biosciences, I was the chief operating officer. And then the same thing was true at MJ GeneWorks. I was the president and CEO. I gotta say, this is not my goal. Okay? These were really stressful jobs. I lost years of my life on these jobs, and I was not kidding. I would walk in Palo Alto, where Cell Biosciences was. I used to walk to Starbucks, and I would walk those tree-lined streets, and every day I would go to Starbucks at the same time, and I passed a mailman. And I was envious of that mailman in his shorts in Palo Alto every day. It was not a fun job to me. As they say, the lead stallion has a short life. I, you know, deliberately demoted myself. I'm having a heck of a lot more fun at my sweet spot developing products than I did as a top executive running a company. It's just be careful what you ask for. If you get that promotion, it comes with a lot of responsibilities, and those responsibilities you will lose sleep. They will raise your blood pressure, and you will die younger. Okay, so final thing, some book recommendations. These are 3 of the most useful books I could think of, okay? And the top one is by far and away the best. You can buy it for like $2, probably less at a used bookstore. How to Make Friends and Influence People. It's like from 1937. Awesome book, especially for me. And everybody I've written it to has come back to me and said, you really probably can get the pitch in the first chapter. But it is so worthwhile reading. You may sit there and say, obviously, well, my son, my eldest son, who's an entrepreneur, he came back and said, I don't do any of that. But he's right. You got to do that. Okay. 101, I just put 1,001. I made that title up. I have a book on my shelf called 501 Interview Questions. Grab one of those books, read the questions, and come up with answers, right? They all look the same, and they all are going to give you the same questions, just resaid. There's really about 20 or 30 questions, just said different ways. Be ready to answer those questions. And there's like a bunch of books like this. It doesn't matter which one. It doesn't take rocket science to write one of these things, but it will prepare you to get a job, and you will do better in an interview. There's also some things out there that on the web about power words and stuff like that. Get familiar with the concept of power words and how to use them. They're really useful in dealing with executives. The Visual Display of Quantitative Information by Edmund Tufte. This guy is just awesome. He gives talks across the country. You can apply to take the course. I think the books come for free if you take the course. Grab a book, probably, there's probably one in every building here or something. There certainly is one in every company I've worked for. It's a fantastic book. And it's a common problem that we get people out of academia who really, at the end of the day, every day I look at a box plot. I don't know if you know, or sometimes called a whisker plot. It's divided into quartiles, if you don't know what it is. But it only is representing 3 data points. It just drives me nuts. Whack 'em. The visual display of data. It's all about your ability to convey a message in a concise way. Something I didn't necessarily do with fumbling with my slides over there. It just becomes a distraction. It's really about, this is the data I want to convey. This is the most effective way to convey it. Learn how to do that. Learn how to write an executive summary, and the display of data, which is your job, right? It's not really useful to do a fantastic experiment if you can't clearly explain the results. Okay? Very, very good book and a wonderful read, and the examples are awesome. It's really, really a joy. And he's wonderful to listen to. Of course, he hates PowerPoint, cannot stand it. Thinks that it has homogenized data display, and he's absolutely right. But PowerPoint is an incredibly powerful tool for me. You notice I didn't go crazy with the animations and stuff because you only use it to try to make points, right? It's not to show off how good you are with PowerPoint. Okay? Right on time. Thank you very much. Half hour, huh, for questions? All right. Actually, I talked for an hour and a half. I was over time. Go ahead. Thank you. Sacrifices. Well, I would say the biggest sacrifices are from my wife and kids. Nobody should have a child while they're a graduate student. Don't recommend that at all. And following me across the country greatly stifled her career. She had this great job at Cornell. It was wonderful. She never worked again. And because, I'd move, I'd go away on long trips. It's just really kind of a tough thing for my wife to do. Like I said, I've already sacrificed years of my life, probably. Yeah, I ask myself that question sometimes. It depends on what phase. Like right now, it's just such a pleasure to work at Life. And on the project, I'm interesting because it is one of these places where my expertise in enzymology, molecular biology, computer science, engineering, they all kind of come together in that sweet spot that I love so much. And when I'm working on a project like that, it's great. But there were times when there would be people in my office crying, and I just did not want that. Wondering, I did not get my PhD to do that. So if you manage to get into those right, I recognize I was going in the wrong direction and go in the right. When I was going in the wrong direction, yeah, I didn't. Very sweet part of your life to kind of give up. I urged him to go down the engineering because I think engineering is a place where you can get the salary and everything without having to spend so much time in school. My PhD was not a fun PhD. Loved the people and my professor, but it lasted a really long time. And 6 years was a sweet time of your life, from 21 to 27. It's just. Mm-hmm. So I personally, when I ran 2 HR departments, I never used a headhunter, ever. Our company only we hire what they call talent acquisition professionals. And they're good, aren't they? They're very good. Huh? Yes. They're exactly the same thing. And so big companies do it because it's all about, Adam Smith, right? It's all you do this and do it really, really well, and that's what they do. It's expensive to use a headhunter. It's expensive to hire people who have temp jobs and hire them away from whatever. I like these temp companies, by the way. They're very useful because there's lots of times I have projects that have 3 to 6 months windows. I know I'm not going to need a person for the whole time. I like hiring people from temps, and it's very often worthwhile hiring those temps afterwards because, man, that's the best interview you can get, right? If somebody works for you for 6 months, I can't tell you the number of interns I've hired. You get a chance to make an impression. So yeah, it depends on the company. When I'm given a choice, the answer is no, because I spend the company's money like it's my own. Thank you. Intelligence and attitude. Really hard. Depends. When things are going well, it's easy, right? You get excited to go to work. I'm very tired by Friday. I only do a half a day on Friday, but I do work Saturdays. And so right now I'm working about 60 hours a week, 57 or 60 hours a week. By the end, I'm really tired. But it's funny, one day off on Monday, I am so psyched to go in. And part of it's my own temperature. In that I'm like that kind of naturally. I'm an extroverted person. I'm very comfortable speaking. My mom used to say I like the sound of my own voice. And but there are many times where I know I have to do it. It is my job to be like this. It's practiced and helped make me successful.
Like postdocs.
5.
So how do you what was it like for you when positions that
So when I actually applied for the job that I took, I was just I would look through science, and if I saw a job, I would apply for it. When my wife became pregnant, then I got serious and started doing 10 at a time. And the job, I ended up getting not a lot of resumes out before that happened. So like I said, I did a postdoc. I didn't want to do a postdoc. I did it for my wife. And when I chose a postdoc, I chose one that was completely different. It was bad to be at the same university, right? They say don't do that. But I knew that if I jumped to something completely different than my training, it would be a real growth experience, and it would enhance my likelihood of getting a job, and I think it did. I'm not sure I answered your question, though. it was different. I mean, in some ways where you are right now, it's one of the easier jobs to get, right? Because you have a job, you have a great narrative, and it's very appealing. And there's a lot of Stanford people out there that are, going to make that one-on-one connection to you. So, I think if you start putting out, and the unemployment for us is, sub 4% nationally. It's very likely that you guys are going to, and that 4% is probably 0.4% for Stanford grads, right? For Stanford postdocs. So you're going to get a job. You actually should think and take your time and get the right job, right? Maybe the thing is really exciting to you, and it is the very first one. But I'd actually be relatively selective, even in today's environment. If you see a company doing stuff that you really want to do, you need to call anybody you know and try to make a connection. Find somebody within that company that you can kind of try to make a connection with and stuff like that. Somebody who already works there, somebody who can take your resume and hand it right to the hiring manager to get around HR. When you really see and you really target a specific place, like I kind of did with Life Tech, you gotta just go to war, right? Build a strategy and do it. Every single thing. Call your mom. Call your mom's brother. Like anybody who knows anybody at that company. Because you're from Stanford. There's going to be professors that know somebody at Genentech, Did I take, when? I did. I actually did. First of all, I used LinkedIn, and I did have people who I'd met along my career who were at key positions, and I would call them up. Hey, can we go, have lunch in Foster City? I did that probably 7 or 8 times. None of those resulted in jobs, but I absolutely do that. Yeah, so I had a couple of slides on the direction that sequencing was going, but I cut them because my talk was too long. And so here's the vision thing. So it started out, radioactive, then it went to fluorescence more automated with capillaries, and then we went into the massively parallel sequencing that 454 first introduced using massively parallel pyrosequencing. Then Solexa quickly followed. There was actually another company in there, but Solexa quickly followed with a superior technology. It still uses these cameras that cost a real lot of money, and they're the limiting factor on the whole system. They have to scan back and forth, and that duty cycle is slow. And they don't use natural nucleotides. They use dye-labeled nucleotides. And those are expensive to make. In a price war, the semiconductor chip, I mean, our detector is not a $20,000 or $30,000 camera, or in the higher end machines, $60,000 cameras. Ours is a chip you throw away. And the data is basically already on the computer. And it really becomes a question of bus speeds. How fast can you pull data off and analyze it more than anything else? It's just such a cheaper technology. In the end, it will win, because it is incredibly cheap. The thing is Illumina is about 5 years ahead of us, and our entry into the market is not as accurate as they are, but it has other specs that are better than they are. So we have a very good foothold right now. And if anything, our problem is that we're delivering improvements so fast that it causes confusion in the marketplace. Which kit do I use? The 100 kit, the 200 kit, the 400 kit? We have 2 different platforms on there, 3 different chips. Illumina's got a single module that they plug in. It's a mature 5-year-old technology. And you got to and they're a little more accurate than we are. We're a lot cheaper. I mean, we have these pluses and minuses that have us have a very nice foothold in the company, in the planet. Now, it's certainly growing much, much faster than they are. So our thing right now to catch up is to now just simplify everything even more, to get us down to just a couple of kits, you know, a couple of key applications to really focus things down. We need to become the most accurate system on the planet. And I don't think that it isn't going to take us, you know, a year. It's just happening so fast. It's just that, you know, if you're in the clinic and you start doing a bunch of science on one of our boxes and we come out with a new improved kit, you can't really use that because you validated everything in the clinic for this particular kit. So there's this constant, we're growing fast, we're bringing something new into the market, and that means it's changing and advancing really, really fast, but, you know, that's a burden that customers kind of have to face if they decide to go with us right now. It is a cheaper technology, and it's going to be the most accurate technology, and it's going to win in the end, largely because of this. The fundamentals of the platform are superior. What comes next? So the hot thing in the field is, well, it's single molecule, which I talked about before. That's another one. Now, the limitation there is when you're watching DNA being made, you have a limited field of view. You cannot move your camera. You'll miss it. So the problem, and things happen fast, right? You're talking about cameras with frame rates on 10 milliseconds, 10 milliseconds frame rates, really, really fast. So these cameras, because they're collecting so much data, actually have very small fields of view. Really, if you want to sequence a whole genome, you need to have a big field of view. You need to view an entire slide, you know? And so that camera does not exist. It won't exist for another 5 or 6 years. You need some sort of fundamental change. And the accuracy is too low. So the next thing that people are going, but I love it. I mean, it's a great technology or everything, and it's the vision of how things will be done someday. It's just that that world is the difficult one to address. Single molecule is stochastic. Redundancy is your friend, but it's single molecule, so there's lots of problems with it. The next thing that's coming down the pipe is something called nanopores. So the idea here is that you have a piece of DNA on a silicon chip, and you just feed it through, and it sequences. All right? Ideally, this has no biology at all, and the speed is lightning fast, as in, you know, sequence the genome in a few minutes, as fast as you can feed your DNA through that little pore. That's not what's coming to market right now. What's coming to market are things with biological pores on membranes that have a finite shelf life, and they have the same kind of stochastic problems that I was talking about before. When you have these electric fields and you're trying to measure which nucleotide is going past, you have electric noise, flicker noise, and stuff like that, that are going to make that even farther out in the future. 454 is dead. We killed it. We have all the advantages of 454 at a fraction of the price and 3 times the throughput. So they're on their way out, and Roche has made a strategic retreat from that market. They're no longer advancing on that. After they failed to purchase Illumina, they basically have said, we're out. So, and that will happen, you know, that's what's slowly happening. Capillary, a lot of sequencing. I would gladly live on the profit from capillary electrophoresis sequencing, because there's plenty of it still going on today, because there's certain things that it does better than anything else. So these other things will always exist at some level, but you know, I think 50 years from now, you'll have some field device where you go out there and to a post office or whatever, and it's has no biology at all. It, you know, sniffs the air for DNA, and it sequences it like a tricorder, you know, faster than your iPad kind of thing. That's kind of the direction we're going with sequencing. So it's really exciting to work on Ion Torrent, because the big thing is when you're talking about a machine that costs less than $50,000, you're not talking about special grants. And it fits, you know, it's quite literally the size of a printer. You know, this completely changes things. You don't need a core facility doing everybody's sequencing anymore, because it's, you know, it's so cheap. You can have one in your lab and get it done whenever you need. And now people can do massively parallel sequencing for nothing, for so much less than it ever was before. And that's just going to keep happening. It's just going to become like an electrophoresis box. Everybody's going to just have it as one of the tools that are in their lab to just do basic science. So I am definitely not the entrepreneur. You know, it's very difficult. So Life Technologies, I think, has one of the most powerful business models. And if you're an entrepreneur, you love Life Technologies. Everybody kind of picks on them a little bit. But what is their business model? They acquire late-stage startup companies. What that means is there's 10 companies out there, all of them getting VC money, venture capitalist money, with their strings attached. But only one of those is going to make it. Only one of those is going to successfully bring, lots of people have cool technologies. That doesn't mean that they're going to be successful in the marketplace. It doesn't mean that that technology is going to be better or cheaper than, it works. It may work fine. That doesn't mean it's the best way to do something. Sometimes sequencing isn't the best way to do something. So you go out and you see you let the market decide, and then you pay for the top. You pay the top price. You pay more for that security. But startups, if you bought by Life Technologies, that's a touchdown. You won. You cash out. Because they pay good money. I mean, for Ion Torrent, it was like $750 million. And they had sold like 10 boxes. Solera, when Illumina bought Solera, had sold 13 boxes. $600 million that Illumina got for that. So you see the pattern here? There's 100 Genesis out there developing sequencers. It is. There are quite literally 50 nanopore companies out there right now. There's one with the name Nanopore, but I don't know that they're going to win. In fact, Illumina had an investment in Oxford Nanopore and has now withdrawn that investment in that company. So, you know, I don't know. I mean, they gave a great speech at Marco Island a couple of years ago that really put them on the map, lowered Life Technologies' stock price for about 1 week. But, you know, 2 years later, there's nothing on the market. And they're a long way, I think, away. It's tough problems, I think, that Nanopore has to solve. So you see you can invest in those things, help, you know, you can be a VC, but that's really the playground. You see who's actually going to win, and then you buy them. And it's great for the startup because the startup now quickly gets worldwide distribution and an awesome, highly trained sales force. You can grow that product so much faster if Life Tech buys you than if you try to grow it on your own. So it's a win-win situation. I think I actually like that model the best, I think, of all of them, personally. Shh, you're not supposed to tell them. Yeah, so this is the thing about Joe and Jane Benchtop when they do their first massively parallel sequencing. They get a ton of data, and they just don't know what to do with it. They don't know how to fish their thing of interest out of it. So I mean, as the developer of the sequencer, we have right of first refusal. In other words, we should be holding your hands and enabling you to do that. In fact, our instruments are primarily like simple fluidics and a couple of circuit boards, and then a computer chip. The real power behind it is the supercomputers that we stick inside the boxes. They're really bleeding edge bus speeds, really, as soon as something new comes out, it's in one of our boxes in a commercial product, really quickly. The compute problem is a problem that, you know, people need to address more. Fortunately, Moore's law is still cranking along. You know, every year these computers get faster and cheaper, and it's coming to us. The only thing is the growth of sequencing is actually like several fold faster than Moore's law. So, you know, we're a little ahead of them right now, but the compute is, I mean, the farm we have in our facility, I have 70 PGMs. these small sequencers. There are 18 protons in my lab. Those things, I mean, the compute farm that I have is, a couple $1 million. That's not what, but when we deliver a PGM, it comes with a box that is capable of doing all the computing you need to use that instrument. If you really crank that instrument, you're going to fill up that hard drive pretty fast. You're going to want to move it out and really get down to the smaller amount of data. Small amount of data. No, yeah. So the data analysis problem is a different kind of problem, and there's a number of ways to address it. I'm certainly not an expert at it, but you basically would like to have another torrent box next to your PGM to just kind of do that if you possibly can. We certainly sell those, but I don't really know much about them, to be honest with you, because my compute farm is completely different, a different model. The proton's a little different in that it comes with an amazing computer inside the box, and then there's an external box that's pretty good supercomputer. But again, I think if you crank that thing as fast as it's going to go, you're going to have that problem there as well. It depends on what you do. So cloud solutions are the thing of the day. The only problem is, if I have a one terabit file, it's going to take a long time to deliver that up to the cloud. So, but sequence files themselves can fit on thumb drives. Depends on what you're trying to do with it. The idea is to get from the raw data to a sequence file, BAM file, or something similar as soon as possible. And then those files are a reasonable size, and people are used to moving those around quite a bit. It's a bit of a shock when somebody gets one of these boxes dropped in their lab and they realize, I did a run and it takes me a week to figure out what the heck I did. And what we're seeing right now is a wonderful trend in that we can see our customers have become accustomed to this. They've now solved those problems, and we're seeing the throughput of reagents on our instrument just skyrocket now. People have figured out that, Joe and Jane Benchtop have figured out how to solve these problems. I don't know the solutions myself because I don't deal with them. You'd have to contact a sales rep. I'm sure they have a better answer than I just gave you. But they've clearly figured out how to solve this problem because they are now sucking through juice like we can barely make. So it's definitely happening. But it is, do you do sequencing? Yeah. Metagenomics. Love those projects. You want long reads like crazy, don't you? Yeah, assembly is one of the most computer demanding applications. We don't even do it in our house. De novo assembly is really compute intensive. Yeah, I got to tell you, Santa Cruz is probably the best bioinformatics school in the country, I would say. But Stanford's right up there. You guys you're good at that kind of stuff. Thank you very much.